Hi all you sake lovers out there, and welcome again to Sake Science, where I delve into the molecular mysteries of one of Japan's signature alcoholic beverages. This time I'm starting a three-part series on a class of compounds that really distinguishes sake from other beverages like wine and beer. They contribute to some of its distinctive flavors and aromas, and also contribute to the ways in which the quality of sake can degrade over time. So join me as I take a look at sake from the perspective of amino acids. Like I said in the intro, I'm dividing this series into three parts: the effect that sake has on the taste, aroma, and quality of sake. They're significant in each of these areas simply because they're present in so much larger quantities than in beer or wine. Although the exact values can vary depending on the specific product, these numbers give an idea of the differences. We can see that sake has about double the amount of amino acids found in beer and 6 times that of the amount in wine. Now, for most people who have learned a bit about sake, amino acids are generally associated with one word: umami. But in fact, the range of amino acids present in sake contributes much more to its taste than just umami. So with this video, I hope to help you understand the ways in which that is true and how it can contribute to your appreciation of sake. First of all, let's review what an amino acid is. An amino acid basically is an organic compound that has two important functional groups: an amine and a carboxylic acid, hence the name amino acid. Now see that R dangling down there on the lower left? That represents other groups that can be present in the amino acid, which gives it its particular characteristics. And depending on the identity of that group, amino acids can be divided into a number of different categories. But for our purposes, there are two categories that I'd like you to remember. 20 to 22 of the amino acids are proteinogenic amino acids. That means that they can be put together to form proteins and also smaller chunks called peptides. The vast majority of the about 500 amino acids found in nature are non-proteinogenic amino acids. And these exist as metabolites of other amino acids, bioactive compounds that can have medical significance, and also signaling compounds that are used between cells, even in our brains. I think most of you watching this are familiar with the fact that the amino acids in sake are ultimately derived from rice, and that the amount of amino acids found in a sake can be influenced by the degree of polishing of the rice. Another factor is the amount of the digestible protein glutenin found in the rice, which can vary by the variety of rice and the growing conditions. When both of these are high, we'd expect the amino acid level of the resulting sake also to be high. But the microorganisms used to make sake also influence the final amino acid level of the sake. The koji, the lactic acid bacteria that's present in kimoto or yamahai sake, and the yeast. I'm not going to get into the metabolism of each of these too much, but here's a quick summary. Remember, koji digests the protein in rice into peptides and amino acids using two enzymes: a protease and a peptidase. And so the resulting amount of peptides and amino acids is high when the expression of these enzymes is high. But what's less appreciated is that koji, lactic acid bacteria, and yeast use these amino acids and peptides as nutrients, especially as a source of nitrogen. Lactic acid bacteria, when present, use these peptides and amino acids to produce metabolites and also other amino acids. And we'll see an interesting way in which this is important in a few minutes. And yeast does the same, while also producing aromatic compounds from amino acids. Moreover, the amount of yeast growth in the moromi and autolysis towards the end of fermentation has a very large effect on the final amino acid level of the sake. The result is a mix of up to roughly 30 different proteinogenic and non-proteinogenic amino acids in sake depending on the specific type. But do all of these amino acids have significant effects on the taste of sake? Actually, researchers have looked into this by taking a model sake and adding and subtracting individual amino acids to it and having panels of tasters tell them whether they could perceive a change in the taste of the sake, and if they could, what that change was. From experiments such as this, 4 amino acids were found to have the greatest effect on the taste of sake. Glutamic acid provides acidity and umami. Arginine provides bitterness and sweetness. Aspartic acid gives acidity and bitterness. Finally, alanine provides sweetness and umami. Altogether, these 4 amino acids give a balance of the basic taste except for saltiness and even a little bit of that. Out of the 4, alanine was found to have the greatest positive contribution to the taste of sake, and it's also the amino acid found in the greatest amounts in sake and in much greater amounts than in beer or wine. The overall effect that amino acids have on sake can most clearly be seen in an umakara, rich and dry type of sake. Here's an example that we had in blind tasting practice at sake school a few weeks ago. As you can see, it's quite dry at +8, and it also has a pretty high ABV and significant acidity, and also a good amount of amino acidity. What's interesting is that in tasting, none of us thought that it was as dry as +8, and most of the students couldn't identify that it was as high ABV as it is. I got closest by guessing 18%. And it's really this high amino acidity that's responsible for this. Imagine, for example, that the amino acidity level was a low 1.0 or even below that. In that case, what would happen would be that the acidity and also the bitterness of the high ABV would really stand out. But higher amounts of amino acids, especially alanine, provide sweetness and umami to balance out the overall flavor and also mask the acidity and the bitterness. But the effect of amino acids on the taste of sake doesn't stop there. They can also help explain some of the perceived differences between sake made with sokujo and kimoto style starters. One thing you may often hear about the difference between these two types of sake is that kimoto has more amino acids. But that's not actually true, and the reality is more complex. Researchers have actually looked into how peptides and amino acids differ in sake made in exactly the same way, except for the type of starter. Here's a graph that shows the changes in the peptides and amino acids in the moromi main fermentation mash with sokujo in red and kimoto in green. And the 2 lines on the top are for peptides, and the 2 lines on the bottom are for amino acids. Just note that I've simplified this data for ease of viewing, and I've also cut out a number of days between the last Tome edition and the endpoint of the measurements, which was day 12. We can see that at the beginning of the additions to the moromi, the peptide levels in kimoto are lower than that in sokujo style sake, and the amino acid levels are higher. But as the yeast propagates in kimoto, it preferentially consumes those amino acids until by the end of the measurements, the amino acids in sokujo and kimoto were basically the same. On the other hand, in the low amino acid environment of sokujo style sake, the yeast preferentially takes up the peptides. And so by the end of measurements for the peptides, those in kimoto were higher. So the upshot is that for sake brewed in identical ways except for the type of starter, the amino acid levels are basically the same. The peptide levels are a little bit elevated in kimoto, but the amount of bitter peptides is actually lower in kimoto than it is in sokujo. And a difference was found in the composition of the amino acids between the 2 types of sake as well. This is due to the fact that amino acids can exist in 2 forms. In eukaryotic organisms like yeast, rice, and you and me, L amino acids predominate. But bacteria can also produce and use D amino acids. The two forms are chemically equivalent except for the way that the parts are arranged around a central carbon atom. This makes them mirror twins, and like your left and right hand, no matter how you rotate them, they can never be superimposed on top of each other. Because of this, L and D amino acids have some different properties, including, perhaps surprisingly, different tastes. And D amino acids are generally found to be more palatable than their L cousins. The amino acids derived from rice are overwhelmingly in the L form, but the lactic acid bacteria in kimoto have an enzyme called a racemase that can actually convert the amino acids between their L and their D forms. As a result, in kimoto-style sake, a notable amount of 3 amino acids in particular can be found in their D forms. And it's also been found that sake with more D amino acids is evaluated more favorably by tasters. Overall, with a more palatable and sweeter amino acid composition and more peptides giving body but fewer of those bitter peptides, Kimoto Zukuri sake is generally evaluated as a bit sweeter than its sake meter value would indicate and as a bit richer than its amino acidity level would indicate. But there's even one more way in which amino acids can enhance the taste of sake and also can explain the interactions of sake with food. And this comes from an understanding of the science of Kokumi. Kokumi is a phenomenon observed whereby the basic tastes of sweet and umami and salty can be enhanced by the addition of certain compounds to foods, even though those compounds have no taste themselves at the concentrations used. Importantly, kokumi is not a taste itself. It's perceived as an enhancement of the onset, the intensity, and the prolongation of these pleasurable tastes, and this occurs through special receptors in the oral cavity that are distinct from the receptors for sweetness and umami. There are a few potential kokumi compounds in certain types of sake, but one that's received special attention in recent years is the non-protein amino acid ornithine. This amino acid can be slightly elevated in kimoto zukuri sake, and researchers have also isolated yeast that produce ornithine in concentrations that are close to those observed as having kokumi effects. An illustration of ornithine's kokumi effects can be seen in experiments where mice were given a choice of 2 water bottles to drink from. Given a choice between plain water and water with a small amount of ornithine added to it, the mice didn't prefer one over the other, which shows that ornithine itself doesn't have any taste. But when presented with a mix of monosodium glutamate, sucrose, and salt in water, or the same mixture with that little bit of ornithine added to it, the mice preferred the second one with the ornithine added almost 3-to-1. Researchers have found similar positive responses by human tasters to ornithine, and they've even potentially identified a novel receptor responsible for this effect. So I think we'll be hearing more about this and other kokumi compounds in sake in the future. So here's where this all ties into the interaction of sake with food. It's often said that sake enhances the taste of food. But with the research that I've done for this video, I actually think the formulation should be the other way around. That is, that food enhances the taste of sake. Nowhere is this more apparent than the interaction of cheese with sake. Cheese, especially fermented and aged varieties, is a concentrated source of potent umami compounds such as nucleotides and glutamate, kokumi compounds like gamma-glutamyl peptides, and also salt, all of which are absent in sake. These can enhance the sweetness of sake, as well as the umami of compounds such as certain amino acids and organic acids present in sake. And there can also be a contribution from Kokumi compounds like ornithine and others that are yet to be discovered in sake. So I'd like to introduce an interesting pairing with a very umami-rich kimoto sake from Kurosawa. This is Kurosawa's Hozumi Junmai Kimoto, and it's actually been aged for almost a year at room temperature. And I'm pairing this with the world's very first koji cheese, which is made by a producer in Miyagi. And as you can see, they make it in this donut shape so that I don't know if you can see that in the camera, so that the koji can actually penetrate more easily to the center of the cheese. And what's interesting about this is that it has amounts of glutamate that are 3 to 5 times higher than that found in camembert cheese, depending on the degree of aging. So let's try these together and see how they enhance each other. So first of all, the Kurosawa Hozumi, wow, it's just got this lovely aroma of brown sugar and kind of like citrus, and also that carries over onto the palate. It's actually not so sweet, but the perceived sweetness is pretty high. So again, it's like that kind of brown sugar kind of flavor, even a little bit of maple. But the acidity in the end is just really like mouth-wateringly delicious, very citrusy, almost like a mandarin orange. And this just really lovely, really long, lingering finish. So this is delicious on its own, but I do think that, this kind of sake isn't something that really is initially appealing to people first trying sake for the first time. So let's see how the cheese can perhaps enhance this. So I've got a pretty good chunk of cheese here. Mm. 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 Wow. I mean, one of the things that really stands out for me is the sensation of mouthfulness. And that's actually a really important effect of Kokumi that comes from basically all of the different receptors in your mouth firing at the same time, and so your brain perceives the taste as coming from all different areas of your mouth, not just localized on one part of the tongue or one part of the mouth. And I think that's a really important aspect of the way that sake interacts with the kokumi compounds and umami compounds in food. So I hope from this video you've learned a little bit more about the different ways in which amino acids can affect the taste of sake, and also how they can affect how sake interacts with food. And in my next video, I'll be taking a closer look at how amino acids affect the aroma of sake. So if you found this video interesting, please smash the like button and subscribe, and stay tuned for my next video. Until then, keep exploring sake.